Hi, I'm Sarthak Khandekar and I'm going to talk about CRISPR and cancer. Let's see what CRISPR is. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A palindrome is a sequence of alphabets which read forward or backward reads the same. So CRISPR is nothing but a repeating sequence of palindrome and spacer DNA. CRISPR is actually naturally occurring defense mechanism in bacteria. Spacer DNA acts as the memory of the last attack. What it means is that spacer DNA stores the dangerous bits of the attacking viruses. Every spacer DNA in the CRISPR system is unique. Technologies before CRISPR include ZFN and TALEN. CRISPR was first discovered by Yoshizumi Ishino in 1987 from Osaka University. The function of interrupted clusters repeats were not known at the time. CRISPR-associated proteins or Cas proteins are used to snip the DNA. The enzymes that encode the Cas are always in the vicinity of the CRISPR sequences. Cas proteins are of two types, helicases and nucleases. The function of helicases is to unwind the DNA. The function of nucleases is to cleave or cut the DNA. Now, we know that Cas is like a molecular scissor. Now, how does it know where to cut? So gRNA acts as the locator. gRNA stands for guide RNA. CRISPR RNA along with tracer RNA is the gRNA for the whole CRISPR-Cas9 system. Protospacer adjacent motif or PAM sequences are 2 to 6 base pair DNA sequences immediately following the DNA sequences targeted by the Cas9. Recognition of PAM by Cas9 nucleases is thought to destabilize the adjacent sequence and thus Cas9 can unwind the DNA. Guide RNAs can transport Cas9 to anywhere, but editing occurs only where the Cas9 recognizes the PAM sequences. So far, we have seen what CRISPR is, what Cas is, what gRNA is. But how does CRISPR work? Let's see in detail. As we've seen in the previous slides, that acts like a molecular scissors. CRISPR plus tracer RNA acts like a gRNA or a locator for the molecular scissors. So, how does CRISPR? To understand how CRISPR works, let's see the two cases that we need to consider. One case where the spacer DNA is already present and the second case where the spacer DNA is absent. The case where the spacer DNA is already present is called targeting. Let's see how it works. So, spacer DNA is transcribed into CRISPR RNA. This is how the CRISPR RNA is represented in this diagram. This is the tracer RNA, and the whole system with the tracer RNA and cRNA is the sgRNA or single guide RNA. Single guide RNA and Cas9 protein forms a Cas9 complex. This is the gene that you need to edit. As you can see, the green sequences are the PAM sequences. PAM sequences are generally of NGG, where N stands for any nucleotides and G is the guanine type of nucleotide. So, as you've seen in the previous slide, the Cas complex is already formed, and this is the gene that you need to edit. Now, the gene is unfolded because of the presence of PAM sequences. This unwound DNA is then attached to the CRISPR RNA, that is the exact match for the gene to be cut. So, the cutting occurs at this position, and then all that is left is two pairs of DNA. Now, the DNA can go auto repair, or you can just insert a donor DNA in it for any mutation that you want. This is the block diagram of how it happens. As you can see, this is the tracer RNA, this is the CRISPR RNA, the whole thing is called the single guide RNA. This is the DNA sequence that you want to edit. These are the PAM sequences, and the CRISPR RNA and the DNA that you want to edit perfectly match. They are cut by these cleaves, and then you have a segment, empty segment of DNA which you needed to edit. You can insert a donor DNA in order to prevent the auto repairing of the DNA, which can cause unwanted mutations. Adaptation is the second case that we need to consider where the spacer DNA is not present. In this case, what happens is CRISPR forms a class 1 Cas protein. This class 1 Cas protein takes the virus DNA inside, breaks it apart, copies it into CRISPR system, and now that it has now that it has the the spacer DNA of the virus, it uses the targeting approach, as discussed in the previous slides. So targeting and adaptation work like this, and it's shown in the diagram pretty clearly. The diamond blacks are the repeat palindromic sequences. The square blocks are the spacer DNAs. As you can see, this is the new virus. A Cas9 Cas1 complex is formed. The spacer DNA is extracted from the attacking virus and it's stored in the CRISPR sequence. After the storing of CRISPR sequence, CRISPR RNA is formed from the spacer DNA, and the Cas9 proteins are uh, transcribed with the CRISPR RNA to form a CRISPR-Cas protein, CRISPR-Cas complex. This CRISPR-Cas complex again uses targeting to deal with the attacking virus, as discussed in the previous slides. So, advantages of CRISPR are mainly that since you can edit anything out of DNA, so it can be used to cure many diseases. Not only can it be used to cure diseases, it can also be used to stop the hereditary diseases from transferring to the offspring. Problems that CRISPR system face are basically off-target effects. What off-target effect stands for is in the 6 billion letters of the human genome, there can be more than one match for the spacer DNA or the CRISPR RNA that you want to match. Self-repairing is actually what we discussed before, that when the CRISPR-Cas9 complex cuts the DNA, the DNA goes auto-repairing. It may there are many random DNA floating around the DNA that you want to edit or you want to cut. So after you cut the DNA, all the random DNAs that are already floating around just rush into the gap, and auto-repairing may cause unwanted mutations. Let's see what cancer is. Cancer is a genetic disease. That is, cancer is caused by certain changes to genes that control the way our cells function, especially how they grow and divide. Probable symptoms of cancer involve a lump of mass anywhere in the body, abnormal bleeding, sudden loss in weight, etc. Some common forms of cancer are prostate cancer, breast cancer, and lung cancer. In a way, we can say that if and when the DNA inside our cells gets damaged, our cells can become cancerous. There, there are two types of mutations. Acquired mutations that are usually the most common cause of cancer. These occur from the gene alteration during the lifespan of a person. These are not hereditary, that is they are not passed from the ancestors to their descendants. Factors that cause such gene mutations are tobacco, ultraviolet radiation, viruses, and age. Germline mutations, which are less common, are hereditary. Here, the mutation is evidently present in every cell in the body of the person, including the reproductive cells of the person, irrespective of its gender. And since the gene alteration affects the reproductive cell, this mutation is from one generation to another. There are three types of genes that cause cancer. Tumor suppressor genes. These are the protective genes that usually control the generation or expansion of tumors. When a tumor suppressor gene is altered, cells tend to grow abnormally and may finally result in a tumor formation. Examples of tumor suppressor genes include BRCA1, BRCA2, P53, etc. Oncogenes encourage cell multiplications and tend to resist cell death, sometimes eventually leading to cancer. DNA repair genes fix mistakes made when the DNA is copied. But if a person has an error in a DNA repair gene, these mistakes are not corrected, and then they become mutations, which may eventually lead to cancer. This is especially true if the mutation occurs in a tumor suppressor gene or oncogene. Let's discuss about a paper that discusses about target therapy in treating bladder cancer. Conventional therapy for treating bladder cancer include chemotherapy and radiation therapy, both of which focus on mass cell killing without a specific target. Targeted therapy only kills or destroys those cells which are the cause of the disease and leaves the rest of the cells intact, even the rest of the cancer cells intact. So, Eugene Liu et al. In their paper synthesizing AND-gate genetic circuits based on CRISPR-Cas9 for identification of bladder cancer cells, developed an AND-gate to target only the bladder cancer cells and leave all the other cells intact, even the other cancer cells. A vector encoding a apoptotic apoptotic or cytotoxic gene is driven by a cancer-specific promoter is used to kill only cancer cells in which the selected promoter is highly active. Promoter highly active in cancerous cells and undetectable in normal cells is chosen for this strategy. The two chosen promoters are cancer-specific promoter, human telomerase reverse transcriptase, HTRT, and bladder-specific promoter, human uroplakin 2 gene, HUP2. As you can see in the figure, this is the Cas9 protein that is shown in cyan color, is targeted on the LACI DNA and designed by the sgRNA consisting of a guide sequence shown in the dark blue sequence here. The sgRNA pairs with the DNA target directly upstream of PAM sequences, and the Cas9 proteins directly cuts this sequence at this position, leading to development of two dsDNA, and then you can then auto repairing occurs, or you can just add donor DNA inside to reduce the chances of mutations, unwanted mutations. To allow sgRNA to rest in the nucleus, products of sgRNA genes are in many cases transcribed from Pol3 promoters. In this case, a Pol2 promoter is used. Pol3 promoters in most cases do not unveil tissue specificity, unlike Pol2 promoters. So, if you want sgRNA to rest in the nucleus in the presence of Pol2 promoters, one of the solutions is to prompt the RNA molecule with ribozyme sequences at both the ends of the sgRNA. This figure shows the same. This diagram shows the sequence and cleavage mechanisms of sgRNA expression vectors. The designed sgRNA sequence is inserted between the two ribosome sequences, and these two sites are the cleavage sites. And after the cleavage, you get the sgRNA as shown in the figure. These are the AND gates that they constructed in the paper. Okay, for testing their experiment, they used these types of cells. So they used 4 types of bladder cancer cells, that is T245637 SW780J82, immortalized cell 293T, cancer cells HeLa A549 PC3, germ cells GC1 and GC2, and primary fibroblast. So according to their experiment, the circuit should only be activated in this bladder cancer cells, these 4 bladder cancer cells, and that's what they showed in their paper, that it did activate for only these 4 bladder cancer cells. So let's see what the parameters they tested were. Specific induction of apoptosis of bladder cancer cells. So as shown in the figure, there are 4 circuits that are basically used for them. So they use SC1, SC2, SC3 and SC4. These are the circuits that we are going to see in the parameters tested. So they used SC2 circuit to check if it can selectively induce apoptosis of bladder cancer cells by regulating expression of human BAX gene. The circuit selectively increased SBAX protein expression and triggered apoptosis in bladder cancer cells without affecting the other cells. Let's see what specific inhibition of growth of bladder cancer cells did. SC3 circuit was used to check if it can control cell proliferations by regulating expression of P21 gene. Specific inhibition of migration of bladder cancer cells. It was found that SC4 selectively increased E-calorin protein expression and showed a more robust inhibition of migration in bladder cancer cells. So all the 4 circuits were pretty good in showing the results of accurately targeting only the bladder cancer cells. C et al. In their paper RNAi-based logic circuits for identification for specific cancer cells established a cell classifier that recognized 6 different HeLa cell-specific microRNAs and only activated apoptosis of HeLa cell in all the tested cell lines. This work shows the conditional regulation due to RNA and also strengthens the possibility of synthetic circuits in cancer therapy, as they have used in AND circuits in this paper that I've discussed before. An additional system that focuses on alike principles is the dual promoter integrator, which incorporates information from 2 cancer-specific promoters to convey anti-cancer gene. This model can be utilized in order to recognize and destroy cancer cells with high targetability. So, the conclusion is, gene editing can be easily done using CRISPR. provided that you have an accurate SGRN. And if you want if you want a perfect results, you need to minimize the problems that CRISPR faces, like off-target effects and self-repairing. Since you can essentially achieve anything by CRISPR just by editing the DNA, and if you have the accurate SGRN sequence, so I would say CRISPR is pretty dangerous. These are the references that I used and studied about and prepared this presentation. Thank you for listening to me.